Hello. So today we're going to be talking about hereditary prenatal development and birth. My group is composed by Armando Piedra, Gabriela Piedra, Montserrat Macia, Isabella Gianni, and I, Shivani Lokani. These are the topics that we're going to be covering in today's presentation. So, genes are a specific sequence of nucleotides and are recipes for making proteins. They're also located on chromosomes, and there are an estimated of 20,500 genes for humans. Normal human cells contain 46 chromosomes in the nucleus of the cells. Mitosis is defined as the cell's nucleus making an exact copy of all the chromosomes and splitting into two new cells. In meiosis, the gametes chromosomes duplicate and then divide twice, resulting in 4 cells containing only half of the genetic material of the original gamete. Of the 23 pairs of chromosomes created at the conception, 22 pairs are similar in length. These are called autosomes. The remaining pair or sex chromosomes may differ in length. If a child receives a combination of XY, the child will be genetically male. If the child receives a combination of XX, the child will be genetically female. So genotypes and phenotypes. Genotype refers to the sum total of all the genes a person inherits, while phenotype refers to the features that are actually expressed. Since genes are inherited in pairs on the chromosomes, we can have either homozygous or heterozygous. Homozygous is a genetic condition where an individual inherits the same alleles for a particular gene from both parents, while heterozygous is receiving a different version of the gene from each parent. Some genes are dominant. This means that they express themselves in the phenotype even when paired with different versions of the gene. For example, facial dimples, curly hair, normal vision, and dark hair. Recessive genes express themselves only when paired with a similar version gene. For example, red hair, being nearsighted, and straight hair. Alleles are different versions of a gene. So, polygenic are the result of several genes, while incomplete dominance is when the dominant gene does not completely suppress the recessive gene. An example of this is sickle cell. To inherit the disorder, a person must receive the recessive gene from both parents. Those who have inherited only one recessive gene are called carriers and should be unaffected by this recessive trait. Monozygote or identical twins occur when a fertilized egg splits apart in the first 2 weeks of development. The result is the creation of 2 separate but genetically identical offsprings. However, if 2 eggs or ova are released and fertilized by the 2 separate sperm, the result is dizygote or fraternal twins. These 2 individuals share the same amount of genetic material as would any 2 children from the same mother and father. So, genetic disorders. Most of the known genetic disorders are dominant gene linked. Examples are Tourette syndrome and Huntington's disease. Recessive gene disorders such as cystic fibrosis and sickle cell anemia are less common. Some genetic disorders are sex-linked. The defective gene is found on the X chromosome. Males have only one X chromosome and are at greater risk for sex-linked disorders due to a recessive gene. For females to be affected by the genetic defects, they need to inherit the recessive gene on both X chromosomes. But if the defective gene is dominant, females can be equally at risk. Here we can see some genetic disorders, as I was talking about: sickle cell disease, cystic fibrosis, PKU, Tay-Sachs disease. Here we can see Huntington's disease, Tourette syndrome, and achondroplasia. Some sex-linked disorders can be Fragile X syndrome, hemophilia. So chromosome abnormalities. It occurs when a child inherits too many or too few chromosomes. The most common cause of chromosome abnormality is the age of the mother. This is because as the mother ages, the ovum is more likely to suffer abnormalities due to the long-term exposure to environmental factors. A very common chromosome abnormality is on pair 21, also known as trisomy 21 or Down syndrome. And this occurs when there are 3 or more 221st chromosomes. And a person with Down syndrome typically exhibits an intellectual disability and possesses certain physical features such as short fingers, toes, folds of the skin over the eyes, and a protruding tongue. When the abnormality is on the 23rd pair, the result is a sex-linked chromosomal abnormality. And a person may have XXXY, XYY, XXX, or XO. Two of the more common sex-linked chromosomal disorders are Turner syndrome and Kleffner syndrome. And Turner syndrome occurs when part of or all of one of the X chromosomes is lost and the resulting zygote has an XO composition. This usually occurs in one of every 2,500 live female births, while Klinefelter syndrome results when an extra X chromosome is present in the cells of a male and occurs in 1 out of 650 live male births. Genetic counseling is a service that assists individuals identify, test for, and explain potential genetic conditions that could adversely affect themselves or their offspring. The common reason for genetic counseling include family history of a genetic condition, membership in a certain ethnic group with a higher risk of a genetic condition, information regarding the results of genetic testing, including blood tests, ultrasounds, and learning about the chances of having a baby with a genetic condition. If the parents are older, have had several miscarriages, have offspring with birth defects, experience infertility, or have a medical condition. So now Gabby is going to be talking about her topic.
I'm going to continue the presentation talking about behavioral genetics. Behavioral genetics is a scientific study of the interplay between the genetic and environmental contributions to behavior, referred to as the nature and nurture debate. Behavioral genetics also has a bidirectional interplay, meaning that the environment can affect the expression of genes and or that genetic predisposition can impact a person's potential. An example of this is a person who has sickle cell anemia can trigger a sickle cell crisis when exposed to oxygen deprivation. To continue behavioral genetics, we can see that the environment and the genotype interact in several ways. The first being passive genotype environment correlation. This occurs when children passively inherit the genes and the environment the family provides. An example is being a genetically athletic family. Second is evocative genotype environment correlation. This refers to how the social environment reacts to individuals based on their inherited characteristics. For example, this can show how your personality affects the way you treat others. The third is the active genotype environment correlation. This occurs when individuals seek out environments to support their genetic tendencies. An example is someone who is musically inclined and good at music, surrounding themselves with an environment that favors these abilities of theirs. Then we have prenatal development. Prenatal development can be divided into three periods. First is the germinal period, the embryonic period, and the fetal period. The germinal period lasts 14 days from conception to implantation of a fertilized eggs into the lining of the uterus. Once a single sperm has entered the wall of the egg, the wall becomes hard and prevents others burns from entering. Once the sperm is entered, the tail breaks off and the head, which contains the genetic information from the father, is fertilized. Unites with the nucleus. Here, the zygote containing the combined genetic information from both parents is formed. The nucleus contains the genetic information from the mother. During this time, mitosis occurs and cell division begins. After just 5 days, there's 100 cells, now called the blastocyst, with an outer and inner group of cells. The inner group of cells or embryonic disc becomes the embryo, while the outer group of cells becomes a support system. This stage ends when the blastocyst fully implants into the uterine wall. This is a fragile process because fewer than half of all zygotes survive beyond the first 2 weeks. Then we have the embryonic period. This starts in the 3rd week. Here, the blastocyst has implanted the uterine wall, which results in a multicellular organism called the embryo. Blood vessels begin to grow, which leads to formation of the placenta. Growing during the prenatal development occurs in 2 major directions. First is from the head to the tail and from the midline outward. The head develops in the 4th week and is the precursor to the heart beginning to pump. If gross chromosomal abnormalities are present, the organism fails at this stage. So at this stage, many mothers do not know they are pregnant, and the embryo can be exposed to many harmful substances such as alcohol and/or drugs. By the end of the stage, the embryo can move and respond to touch.
The fetal period. From week 9 until birth, the organism is referred to as a fetus. Throughout the stage, the mature structures are developing. The fetus has all of its body parts by the 3rd month. In the upcoming weeks, hair, nails, teeth, and the excretory and digestive systems will keep on developing. During months 4 to 6, hearing develops and the eyes become more sensitive to light. The respiratory system keeps on developing, and reflexes such as sucking, swallowing, and hiccuping develop during month 5. Patterns of sleep and wakefulness are present at this time as well. The age of viability is reached at about 24 weeks. Most of the neurons in the brain have also developed, and the glial or nerve cells that support neurons continue to develop. At this point in time, the fetus can feel pain. Between month 7 through 9, the fetus is mainly preparing for birth. It is utilizing its muscles and its lungs to start to expand and contract. The fetus gains around 5 pounds and 7 inches during the final trimester, and during month 8, a layer of fat that works as insulation and aids the baby in regulating body temperature post-birth develops under the skin. By the 37th week, all organ systems are developed enough for the fetus to survive outside the mother's uterus. The fetus continues to gain weight and grow in length until about the 40th week. Prenatal brain development. By the end of the 3rd gestational week, 2 ridges appear along the neural plate, forming the neural groove, followed by the neural tube. The open region in the center of the neural tube forms the brain's ventricles and spinal canal. At the ends of week 8, the neural tube has further evolved into the forebrain, midbrain, and hindbrain. Brain development throughout the fetal periods deals with neuron production, migration, and differentiation. The majority of the 85 billion neurons have been generated, and many have already migrated to their brain positions starting from the early fetal period until mid-gestation. Neurogenesis is mostly completed after 5 months of gestation. Neurons that form the neocortex migrate to their location in an organized way. Neural migration is mostly completed in the cerebral cortex by the 24th week. Neurons begin to produce dendrites and axons